please take it away.
Thank you for the introduction. I would like to thank the organizers for inviting me here and letting me present my work. So as he said, I'm going to talk about this tool called C-State that I've been working on. I call it an interactive epigenome browser, but all the browsers are interactive, right? But why another browser? Because when we were working with many ChIP-seq datasets and we wanted to use the traditional genome browsers, we faced some limitations. For example, many times you want to look at not just one gene, but several genes that you're interested in, right? And they could be scattered all across the genome. And in many of the traditional browsers, you have to actually pan through the chromosome or region and not immediately jump to other loci, right? And similarly, they do not really understand if the what the data sets are. Like, for example, if you have, let's say, 4 different chip sequencing data sets belonging to a certain cell type, to most of the browsers, there are 4 independent data sets. They do not generally realize the fact that they are coming from the same cell type, right? So whenever you want to compare or analyze all of them as a whole, you'll have to always keep in track of how these data tracks are arranged. And there were several other challenges which I'll explain as I'm going through. So a lot of talks were there, and they were saying live demos are a bad thing, but as I've not made any slides, I have no other option. So I'm going to go to the site. Oh, I think I already have it open. Yeah, so this is the website of C-State where you get the tutorial and all of the things. And so this is a fully online tool, which means you can actually load it directly from your web browser. But once it's loaded, it's 100% client-side, which means you don't have to transfer any of your data across the internet. So there's a prominent launch C-State button, which points to the stable version of C-State. But I'm going to go and load the development version, which I've recently updated based on the comments I got from the reviewers. So this is the main page of C-State. You have this button to load your genes of interest, or also called C-State data. It's basically a way to restart your session, last session, which I'll come back to later. You begin by selecting a species of choice. There are some 8 species for now, which support many other builds. I'm not going to show you how to load your data right now in the interest of time. I'm going to go ahead with the example data sets that are preloaded. But one thing I want you to notice is this particular section, which talks about the upstream and downstream flanks. Because C-State, in C-State, we believe in a gene-centric approach. Instead of a chromosomal region, we analyze each gene as a unit. And many times you know that the regulatory elements or elements of interest often lie upstream or downstream to the gene body. So at the very first step itself, you get to decide how much you want to analyze upstream and downstream of each of your genes of interest. Anyway, so I'll go and click this, which says load example data of 4 cell types. So all this data is collected from the ENCODE consortium. This data belongs to 4 different cell types, and oh, that was fast, okay. Last time it took forever, I was worried. So, this is the main pane. Like, as soon as the data is loaded and the files pane disappears, and you have the view pane coming into the screen. And so you can see that it's showing data for 330 genes. So these genes were actually handpicked from various previously published studies, and they have been known to have important roles in differentiation, maintenance of pluripotency, and some housekeeping genes, and so on, okay? So here, the data of each cell type is arranged vertically. Like, you can see that there are columns, and this title explains which cell type it belongs to. All of these are human cell lines. You have embryonic stem cells, GM12878, which is blood cell, HeLa, everyone knows, and K562. So each of these panels is actually a gene panel, and so the gene body is actually represented by this the black bar, and the upstream and downstream flanks are represented with these steel blue bars. And if there are any neighboring genes present within the 20 kb that we chose, or any other flank that we chose, they are also represented by this gray bars. Okay, and so what I've loaded is actually the chip sequencing data for 4 different histone marks. So K27, H3, K27 trimethylation, and K36 trimethylation, K4, and K9. So along with it, I've also loaded the RNA-seq data set of these cell types. So these are the FPKM values, and that is, so by default, they're represented on this gradient. So which shows that, so for example, these values are actually automatically fetched, and they represent the 5th and 95th percentile expression percentile of your data set. So, and where does your gene of interest stand in each cell type? And that's what is represented here. So something on the top means that it's close to 3.2 or maybe more. And if you actually hover on that, it tells you what's the actual expression or FPKM value. Similarly, you can hover on any of these to get the details of these. So these are actually peaks. So you can load the peaks in most of like bed, broad peak, or any format. So the scale is represented in KB. So, and if you click on any gene, it opens this other view, which we call the modal view. And here the conditions are stacked vertically, or different cell types are stacked vertically so that you can compare them immediately. And if you notice, some bars are quite opaque and some are transparent. So this is actually a function of the peak score that was provided. So any peak that had a high score is represented as a more opaque bar, whereas something that is not so good is represented more transparently. Right? And in this view, you can actually see them as exons and introns. So the exons are these raised bars, and if you hover on that, you actually see what exon it is and so on, what's the distance from TSS. So another important design decision that we made was that irrespective of what is the strand of the gene, we have always plotted it left to right, which means even if the original gene was on the negative strand of the chromosome, the TSS was still plotted on the left side. This helps us immediately compare the genes genes on both the chromosomes, and still not get, I mean, you don't have to do this mental calculation of, oh, now the promoter is on this side, and I have to look at downstream to look at the promoter, and so on. Okay, so the plots are interactive. You can zoom and pan, and these are manual zoom controls. I'll not go into this thing. You can actually also download whatever plots you're seeing as an SVG. Okay, and we have a quick search bar which lets you type any gene of interest, and it will immediately show. For example, if I type CBX, it will show me, for example, all these genes which are CBX genes, and you can quickly look at what's going on with them. Okay, so what I really feel sets CState apart from other browsers is the filters panel. So this is a genes filter panel. What this panel gives you is access to a set of filters in which you can set criteria to actually retrieve the genes that match those criteria. Okay, so there are some simple ones, like you have a name filter where you can specify which gene you want, or size filter. Maybe I'll just demo that. So if you want to only analyze, let's say, genes under 10 kb, so you have, it says show genes, which are, you have a set of operators, and you want to say less than 10 kb, and you go and say apply filters. So you can immediately see that out of the 330 genes that I loaded, I don't know if the text is visible, actually, it's pretty small, but 236 have been filtered out, and if you go back, you'll see that the remaining 94 are actually displayed here. But that's a simple thing, right? But we have other filters. So for example, let me clear this off, and we get all the 330 genes back. So the more interesting ones are like these feature counts and overlaps. For example, I go to the overlap filter. So this is a lot more going on in this filter. You can choose which cell type you want to filter that. So let's say I want to filter on human embryonic stem cells. And what this filter lets you do is you can specify the overlap of 2 different features. So features here being the histone marks, and you can ask where all or which all genes do we have that particular criteria going on, right? For example, I want to filter all the genes which have a bivalent mark coming near the promoters, right? So we know that K4 methylation, overlapping K27 methylation is a hallmark of ESL, so we expect some genes to have this thing. So I choose my first feature as K4 trimethylation and second feature as K27 trimethylation. And in this relation settings, you can say it should be present upstream to or downstream to, or it can be overlapping, or it can be either side. So I'll say overlapping, and I want it, so it says distance to with TSS, with respect to TSS, so I say within 5 kb of TSS, both upstream and downstream. And you can immediately see that 170 of these 330 have been removed, and now these are the genes which have a bivalent mark going on at the promoter in embryonic stem cells. But that's not all. You can actually chain these filters, and they will all be applied sequentially, so that you can immediately ask, for example, out of these 160 genes that have a bivalent mark in ES cells, let's say how many of them have retained a K4 trimethylation mark in some other cell type, let's say GM12878. So I click on this, let's say features count filter, it just gets appended to the filters panel, and here I choose my cell type as GM12878, and I want to look for a presence of K4 trimethylation mark. So I say K4 trimethylation, and I choose that the features count, which means the number of times a peak of that particular mark is called, should be greater than 0. And I put the same restriction of TSS, 5 kb upstream and downstream, right? And I apply filter. So you can see that out of the 160, 32 have been removed, and the remaining of them have a K4 trimethylation mark in GM12878, and these are also bivalent in embryonic stem cells. And that's not all. You can apply any number of filters sequentially to, for example, identify genes that go on to become active in one cell state, whereas because K4M3 is an activating mark, or repressive in another cell type. And I'm not going to demo all that because it will take some time. For example, you also have an expression filter. Now you can ask, in these genes, I want to find out which genes are highly expressed. And we know that 3.2 is the 95th percentile. So for example, you can add another expression filter and ask, let's say, how many of these are having a minimum expression of 3.2 in GM12878. And so most of them have been removed, for example. So only 16 of them actually show a high expression in GM12878, despite having, like many other genes having the K4 trimethylation mark, right? So that, more filters are underway, but I guess this is what really helps. Like particularly, people who are comfortable with bioinformatics obviously can get these answers very easily, but people, for example, even in my lab, they are not comfortable using command line tools. And for them, I just load the data set onto this browser and give them, and they can actually play around with it without having to resort to programming or command line tools, right? So maybe I should have retained it. Doesn't matter. So the next thing is it has a plots and analysis section. These feature histograms are not so important. It will let you see what are your data ranges. For example, you can see, the scores here are the UCSC scores, like it has a range of 1 to 1,000. For example, you can see which of your data set has most peaks with a good score. For example, you can immediately see that in K562, K27, M3, most of the peaks are close to 1,000, which means that's a good data set, right? Most of your peaks have high scores, and so on. So that's just more like a data validation or something. What I like is this average feature profile. So what it does is, it takes all the genes that you have loaded, and it calculates the average profile of each mark with respect to the entire gene body. And you can see that, for example, K4 trimethylation, K9 acylation marks go up. So this is the TSS of all the genes. In general, it is peaking at TSS and slowly falling down. And K36 methylation, which is known to mark exons, is generally higher, like it picks up close to TSS, after TSS, and it stays, and then it falls after the TES, transcription end site. What really strikes us is, in embryonic stem cells, unlike other cell types, you can also see a peak of K27 methylation coming up, which is, again, as we know, are bivalent regions. And you can also toggle between all the genes in your data set or also just the filtered genes. So whatever were actively shown after you apply the filters, you can only look at that. Or you can just restrict yourself to the TSS, let's say 5 kb on either side of TSS of all genes. And it will show you that. And you can also look at the gene expression scatter plots. So for example, if you set some filters for your chromatin marks, you can immediately come to this and see what kind of expression patterns are there. All of these images can be downloaded as SVGs. The other section is a table summary, which basically gives you a summary of all the genes that you're analyzing in a table format. You can again choose between all the genes or your filtered genes. The table is interactive and you can sort based on any column. For example, one thing that we regularly use is by sorting on CDS size. So CDS size of 0 indicates that they're non-coding RNAs. So you can look at all non-coding RNAs like that. And clicking on any gene name will take you to the modal view of that. And finally, a lot of things are customizable, like you can all colors and ranges, data sets. So basically, the philosophy was that have sensible defaults, but give enough customization options so that they can choose whatever in whichever format. And you have several download options. You can export a summary file, or you can download all the view panels as an SVG, or the final option is the download C-state JSON file. As I was telling you, if you download that file, you can re-upload that file as the previous C-state data, and it retains, in fact, you can pick it off right where you stop. And you can also share this JSON file with any of the people you want, your collaborators or anyone, and they can load that file onto C-state, and they retain full functionality of it. And I guess that's all. I would stop there, but yeah, so yeah, whatever. So this is still in a very early development, this thing, as you can see. A lot of things can be added, and it would be really good to have feedback.
Thank you very much. This was an impressive live demo. I don't know why you think it's an early stage of development. It worked fairly well. So questions, please.
Hi, great talk, actually. What I liked the most is the fact that, in fact, you destroy the sys view of the classical browsers, and you now have the genes in a kind of side-by-side manner. So actually, one application that came directly to my mind is, of course, to check at coordinate context, like in the case of chia pet or kaya pet, as you call it in English, and high-C data, if we can use coordinate-based information, not gene-centric, but
Okay, so the previous version of it had an option to load a bed file, your coordinates, but I've now removed it to actually sort of stick to the gene-centric approach, but maybe it's, you can always load it as a feature file instead, because it's not limited to ChIP-seq or RNA-seq datasets. You can load anything which can be represented as coordinates as feature files.
And the other aspect that I was also thinking about transcription factor target genes, you know, instead of selecting a group of genes or loading genes, you could decide with a given TS, which in your database you can have the list of different genes that are basically connected.
Okay, so a direct limitation that I can point to you right now is that we have started with a known set of genes, right? Many times you don't want to do that. So just when this was accepted to BioVis, I started a rewrite of this in which you can load the entire genome and all the genes, and all the filters will work. So then what you said is possible. You load a transcription factor data set, and you can set a filter for all the genes that come up positive for that, like that.
Okay. No more further questions?
Okay.
Do you have a poster, by the way?
No.
No poster, okay. Maybe we can find you around the poster session for more live demos. Very impressive. Okay, thank you very much again.